My name is Charlie Nunn. I'm at Duke University in evolutionary anthropology and Duke Global Health. And I also run the Triangle Center for Evolutionary Medicine, which connects the local universities here in North Carolina around evolutionary medicine. And together with a bunch of other centers, we organize Club Ev Med. And we also work closely with the International Society for Evolution Medicine and Public Health to organize this and other events. And I just want to recommend or ask everyone to consider coming to our annual conference, July 8th to 10th in Nashville. You can still sign up and register and be a part of our conference, and also to submit articles to the journals, the Society's journal, Evolution Medicine and Public Health. And I provided some links in the chat. This is Club Ev Med. We actually like to call it Club Ev Med Conversations technically, because we do want to stimulate discussion. We have a short presentation of roughly 25-30 minutes today, and we want to hear your thoughts. There'll be opportunities at a break point in the presentation to share some thoughts, and we really love to see comments and questions go into the chat as the presentation is given. Okay, I'm going to turn things over now to Randy Nesse, who's going to introduce the speakers. Thank you, Randy.
Hi, everybody, and thanks, Charlie. So I'm going to give just a quickest introduction to this. I was thrilled when I found this paper just a few weeks ago for many reasons. First is to get to know Susan again. I knew her from the old days in the evolution and human adaptation program and the evolution human behavior program at University of Michigan, where she took her degree in 1995. I'm going to ask her to introduce the other speakers, since she knows them much better than I do. Jacinta Beener is my great close colleague and friend from University of Michigan as well, who's the last author on this paper. I really look forward to their presentation, but just to frame it slightly, my original research was on neuroendocrine responses to stress and the original work on cortisol and depression and all, and the results were erratic. And I spent much of my career trying to convince people that stress was useful, because everybody who thinks stress is bad for you. And the idea that it might be useful is just counterintuitive for many people. This article, I think, is one of the first to really nail it in a good study in the wild. Fabulous methods. They also use marvelous natural experiments with the El Nino drought. I won't say any more than that because they have lots to say. I'll ask Susan to start off, and before I do, put your questions in the chat or your comments. We're so eager to engage with everybody about this. Susan, over to you.
Yeah, so just a little bit about the co-authors. The behavioral endocrinology component of my 35-year project began with Colleen Galt's graduate work on that, and she was investigating stress. So a lot of this work is building on her thesis work. And Julianne Dam was working with her as a master's student and also joined me to collect samples during this period. And Ashley Mensing and Irene Godoy were both graduate students working on behavioral endocrinology in my project too. So we're the Lomas team, and I run the project. I was the one there during this El Nino drought, who realized that this was a thing we needed to look into. But we didn't have a lab, and so then Jacinta became involved. She agreed to have we'd send the samples there. Then COVID hit, and my team couldn't even go to her lab. And meanwhile, Sophia was in need of a dissertation remake because of COVID and impact on her field site. So she joined us and did a lot of the assays on site in Ann Arbor. And I think that's basically it. That's who these people are. And so Jacinta and I were the co-lead authors, her being the lab person, me the field person. And Irene and Sophia were co-first authors doing the analysis.
Great. Thank you, Susan, and thank you, Randy. Thank you, everyone, for being here. We're really excited to talk about this paper with you all. So our paper is titled Stress Responsiveness in a Wild Primate Predicts Survival Across an Extreme El Nino Drought. Traditionally, there have been two different approaches to stress research. The biomedical field tends to focus on how the stress response adversely affects human health and longevity. And there's a wealth of evidence linking chronic stress with poor mental and physical health. That includes increased risk of anxiety, depression, and cardiovascular disease. The second approach comes from ecology and evolutionary biology, and this examines how the stress response adaptively helps individuals survive and recover from stresses. But this literature is largely based on experiments conducted on lab animals leaving us without a clear understanding of the costs and benefits of the stress response in natural settings. Both approaches focus on the hypothalamic pituitary adrenal axis, or HPA axis. This pathway is highly conserved across vertebrate species and is activated in response to stressors. This includes both acute stressors, like a predator attack, and long-term challenges, like a drought. When activated, the HPA axis increases the production of glucocorticoids, most commonly cortisol in humans. And these are steroid hormones that regulate energy use and storage in the body to aid in survival of the stressor. Typically, we think of a good or healthy stress response as consisting of low baseline levels of glucocorticoids that increase dramatically following the onset of a stressor, and then slowly return to baseline once the challenge has passed. But like any other physiological system, the stress response has trade-offs. So high glucocorticoids are beneficial for immediate survival and recovery. But in the long term, the stress response can negatively impact health as it decreases the resources available for other bodily processes such as immune function. It's this cost to long-term health that's typically measured in biomedical research. So this figure shows how the stress response may become dysregulated over time. At first, the HPA axis successfully returns the body to homeostasis or baseline levels after a stressor. But after many consecutive stressors, the stress response becomes less effective and can ultimately develop a new homeostasis at a higher baseline level. This results in a cumulative toll on the body, which we often refer to as allostatic load or just wear and tear. And there's wear and tear coming from studies in wild primates as well. Most notably from the Amboseli baboon project, Campos and colleagues measured HPA activity in female yellow baboons. And they found that females who experienced high levels of glucocorticoids across adulthood, shown here in pink, had shorter lifespans than females with low levels of glucocorticoids in blue. Survival models specifically predicted that these females would die up to 5 years earlier, which is a long time in a baboon lifespan. So this is a clear example of how sustained activation of the HPA axis causes wear and tear to the body and reduces longevity. These findings also conform to the court fitness hypothesis that you may be familiar with. This describes the negative association often found between cortisol and fitness. And we see lots of other evidence for this in humans, most notably the association between chronic stress and an increased risk of various diseases. There is a causal effect here, and that's the wear and tear of the stress response. So this research has been crucial to identifying risk factors for disease and potential prevention strategies. And it's been particularly useful to explore as a pathway for the biological embodiment of social determinants of health. However, it's also important to remember that cortisol and fitness are both driven by a third variable, and that is the environment. A more challenging environment will result in higher levels of cortisol and decreased fitness. Think of this as comparing populations that experience different levels of predation. More frequent encounters with predators will increase cortisol levels and will decrease the likelihood of survival, simply due to predation risk, not the wear and tear of the stress response. So when we compare across populations that are experiencing different sources of stress, the court fitness hypothesis often reflects a spurious correlation, and it does not help us make predictions about how an individual's stress response helps them survive a particular stressor. To explore the adaptive nature of the stress response, we need to ask, would an individual survive to face the long-term consequences of wear and tear if she did not adequately respond to stressors in the first place? Here, we would actually expect the opposite of the court fitness hypothesis. A larger increase in glucocorticoids in response to a stressor should increase survival of that stressor. Testing this prediction is very difficult, especially in natural settings. When faced with a severe life-threatening stressor, an individual can only respond in one way, and we can't compare an individual to itself in an alternate reality. Instead, we need to take a reaction norm approach. So by measuring each individual's baseline and stress-induced levels of cortisol, we can calculate the strength and direction of their stress response. Then we can examine how the stress response affects survival. Using a reaction norm approach is still very difficult to do in wild primates, as ethically, we cannot design experiments that would expose individuals to the same severe stressor. The study we're discussing today is unique in that we were able to take advantage of a natural experiment. Beginning in 2014, a strong El Niño struck Central and South America. El Niño events typically result in low rainfall across Central and South America, but this particular event was one of the 3 most severe ever recorded, and it caused widespread drought throughout the tropical dry forests of Costa Rica. And it just so happened that in northwestern Costa Rica, there was an entire population of capuchins under study, and they were heavily impacted by this drought. And I'll leave it to Susan to tell you more about these capuchins.
Yeah, so I want to tell you why capuchins are such a good model for studying stress responses in humans and in other long-lived social animals generally. Capuchins have independently evolved many of the traits that are part of the special adaptive complex in humans, the things that anthropologists get excited about. So they're omnivores, they're skilled extractive foragers, they have extraordinarily large brain-to-body ratios, which are surpassed among primates only by humans, and they have very slow life histories. So females mature at around 6 years of age, and this species has a maximum lifespan of 55 years, though it's more like 30-something in the wild. They occupy a really wide range of habitats, and they're very flexible in their diets and in their behavioral generally. Although their mortality rates as infants can be high due to infanticide, in general, mortality rates are quite low. Next slide. So our particular population lives in the Lomas Barbadal site. It's a highly seasonal tropical dry forest characterized by virtually no rain between mid-November and early May, and heavy rains during May and June, and then especially in August to October. There's typically a mini dry season with only very light rain in July. So this graph you're seeing is for Palo Verde, which is a site next door, well, connected to our site. And this does correlate extremely well with Lomas, but there's a longer range of data for Palo Verde. The Lomas capuchins are fortunate to have a lot of permanent streams and rivers in their habitat, which buffers them somewhat from the seasonality. And they spend a lot more time in these riverine areas during the peak dry season. Though my study began in 1990, our work in behavioral endocrinology started in 2006 with Colleen Galt's thesis work, which documented the impact of social and ecological stressors on fecal glucocorticoid levels. Although Colleen was able to pinpoint some social factors that caused spikes in FGC levels, the major ecological factor that she was looking at, which was human-caused habitat destruction, didn't really seem to have an impact. Furthermore, during the first 24 years of the study, we saw very little behavioral, morphological, or demographic evidence that the monkeys were food stressed during the dry seasons, even the particularly extreme dry seasons, aside from the fact that they sometimes spent less time socializing and more time foraging during that transition month from the dry to the wet season, just 2 weeks or so right before the rains came. So we started thinking that capuchins were so behaviorally adaptable that they could overcome any ecological challenge they were confronted with without any undue physiological stress, and that further investment in this topic was useless. So yeah, next slide. Yeah, that's the right one. So just as I'd stopped fecal collection, there was this major El Niño drought that has been mentioned in the previous slide. So for the first time since I started the project, the rainy season was so dry that the leaves of the trees and understory plants withered and dropped from the plants during that veranillo, that mini dry season in July, in addition to the main dry season. And this wreaked havoc with the food supply. The monkeys themselves visibly lost weight. Now, we couldn't weigh them, but I started seeing ribs and vertebrae in the tail, and you can see the really scrawny neck in this monkey. This is really abnormal. I also want you to notice in this photo that there is a baby, dying baby, lying next to this female. So this was the most shocking change. It was in the behavior of mothers towards their infants. Normally, females are so nurturing that they carry a sick baby or even a dead baby for days after death. Even if that corpse is covered with stinging carnivorous wasps that are attacking the mom as she tries to defend the baby, she'll hang on to that and won't let it go for 3 days or so post-mortem. She won't try to pick it up when it's dropped to the, so now she won't, in the El Nino drought, she wasn't even going down to the ground when she dropped that baby. Even if it was crying to be picked up, she wasn't going to get the baby and she'd eventually walk off and abandon it. So this blew my mind. This was so not capuchins. And I realized, oh, we need to collect more fecal samples again. So I told Jacinta about this and she was interested in the positive, the adaptive effects of cortisol. Okay, so next slide. And indeed, in hindsight, we were able to see that the mortality rates for infants and for older females really soared during this 2-year drought. So what you're seeing in this graph is you've got exposures, so females available to die along this axis on the bottom. You've got 3 kinds of dots here. The gray is the normal years, the black is El Nino years, and the orange diamonds are the severe El Nino years. And this dotted line is the predicted mortality rate. So you can see that during this, especially the second year of this severe El Nino drought, we had really high mortality. So now we were ready to investigate the factors that predicted whether females lived or died by using our physiological data. In this species, females remain in their natal groups their entire lives, whereas the males emigrate to other groups. So when a male disappears, we don't know whether he's dead or whether he just is roaming. However, when a female vanishes, we're sure she's dead. So that's why we decided to conduct this study only on the females. Okay, I'm going to pass this along to Irene now, and we can take some questions if there are any while we do this transition.
So could we just have people raise their hands or go ahead and put something in the chat if you have a question? I have a very quick one. What's the N? How many subjects were involved in the study?
We had, what was it?
We'll get there in a second.
Yeah, well,
looked
it's
to me like
28
there were...
females, 14 survivors, 14 fatalities.
Okay, good.
And a lot of poop from each one.
Okay, without any other questions, let's go ahead.
Okay, can everyone hear me now?
Yes.
And can people see if I scroll with my cursor?
Mm-hmm.
Great. Okay. So as Susan pointed out, we had an unprecedented number of adult females that died during this drought. Nearly 1 in every 3 females in our population died during this period. And these are mortality rates that we typically only see in very young infants. So it was quite a remarkable occurrence. This natural experiment afforded us the opportunity to ask whether individuals that survived the drought differed somehow from individuals that died during the drought. Specifically, we wanted to know whether individuals that responded to previous drought risk with a more positive glucocorticoid response were more likely to survive. To do this, we set out to compare fecal glucocorticoid metabolites from survivors of the El Nino drought and compare them with fatalities. For fatalities, we had 14 females with sufficient fecal samples available. By that, we just meant we knew we had at least 10 samples from them. Now, we matched these 14 fatalities with 14 survivors, choosing a survivor match for each fatality from the same social group. Well, actually, sorry, I should point out again that the reason we were hoping to measure these fecal glucocorticoid metabolite levels was so that we could see whether how females responded to prior drought risk predicted the likelihood of surviving. So in other words, were females that showed a stronger and positive response to the drought risk prior to this El Nino, the severe El Nino drought? Were they the females that were more likely to survive? Well, you might be wondering how exactly we measured drought risk. That's a really important variable for us to talk about in this analysis. We used
Sorry,
18
Irene.
Yeah.
The slide's a little fuzzy. I wasn't sure if it was just going to be that first one.
Oh, let me see.
I think it's probably just a slide. Go to the next slide and see if it's also fuzzy.
Is this fuzzy?
It's a little bit fuzzy too.
Hard to read the font, yeah.
Not sure there's anything to be done.
Let me try to do this. Is that better?
Yeah,
Okay, we'll go with that. Thank you for pointing that out.
maybe read the labels more than you would normally.
Okay, good. What we did to measure drought risk was we used 18 years of daily precipitation data from the nearby Palo Verde field station that Susan mentioned. And we used this to generate a precipitation index. In the top blue panel, what you see are 30-day cumulative rainfall estimates across a 9-year time span. You'll notice that this captures the bimodal nature of the rainy season that Susan mentioned earlier, and also the extended dry season where there is very little to no rain. Below that figure, you see a precipitation index that we calculated based on these cumulative rainfall measures. This precipitation index represents the number of standard deviations above or below the historical 30-day mean for each day. From this precipitation index, you can see some patterns. For example, in 2008, that was a year marked with higher than expected rainfall. And importantly, during the El Nino drought, you can see that this period was characterized by prolonged period of below average rainfall, right? Precipitation values are quite negative. And drought conditions were particularly stark in the first half of the rainy season, so much so that it almost eliminated the first peak that is typical of rainfall at Lomas. Now, if we zoom into the El Nino drought, you can see the distribution of adult female deaths during this period. These are represented by gold rug plots at the bottoms, if you can see them here. We can see that most of the adult fatalities from our population happen during drought conditions in the rainy season.
When it was supposed to be raining.
When it was supposed to be raining, and this prolonged period of below, average rainfall during this time. This was when the majority of the deaths were occurring. So now with a way to measure drought risk, we could get back to our question, which was whether responsivity to drought risk would predict survival across this severe drought. Now, before looking at individual reaction norms, we first ran a preliminary model controlling for factors known to affect glucocorticoid levels. These included female reproductive state, age, rank, group size, and time of day. We also included expected rainfall, which is a measure of seasonality, where, for example, a value of -1 represents periods of low expected rainfall, or in other words, at Lomas, the dry season. And for example, values of +1 represent periods of high expected rainfall, or in other words, a part of the rainy season. Now, in this preliminary model, only expected rainfall, female reproductive state, and time of day accounted for variation in fecal glucocorticoid levels. Specifically, females had higher glucocorticoids when they were pregnant, during the dry season as expected and earlier in the day. Based on this, we could design what we called our reaction norm extraction model, where for each female, we could estimate their individual reaction norm to drought risk. This was done by modeling the interaction of drought risk with expected rainfall and including this interaction as random slopes in our model. From this model, we could extract individual reaction norms to feed into a subsequent model, which would test whether reaction norms predicted survival. We were interested primarily in the effect of drought risk during periods of high expected rainfall. Remember that most deaths during the El Niño occurred during the rainy season when there were these heavy, severe drought conditions. So we extracted our reaction norms based on model estimates for drought risk in the wet season. Specifically, we set expected rainfall constant at 1, representing conditions in the rainy season. As you can see, survivors tended to have more positive slopes in response to increasing drought risk compared to fatalities who had more negative slopes. And indeed, our model, we did find strong evidence that a more positive glucocorticoid response to increasing drought risk was associated with a higher probability of surviving the drought. So for example, capuchins whose slopes, their reaction norms were 1 standard deviation above the population mean, they were 2 times as likely to survive compared to females who were 1 standard deviation below the population mean. Now, another way to frame our question using all the data that was available to us was to ask a similar question, but frame it in terms of whether survival of the El Niño drought predicted females' previous response to drought. This could be accomplished by using a 3-way instead of a 2-way interaction to see whether reaction norms differed based on the survival status of females. Now, once again, remember, we're specifically interested in reaction norms to drought risk during the rainy season. Again, we found strong evidence that fatalities and survivors differed in their response to previous drought risk. Survivors, on average, had positive slopes with increasing glucocorticoids in response to lower than expected rainfall, where in contrast, fatalities had negative slopes with increasing glucocorticoids in response to higher than expected rainfall. Now, it's important to take a moment to keep in mind that these results don't imply HPA dysregulation in the fatalities. This is because both survivors and fatalities exhibited the expected seasonal increase in glucocorticoids during the dry season. But also importantly, during times with seasonally appropriate rainfall, so rainfall typical for the year, capuchins and that survived and those that died, they exhibited similar glucocorticoid levels. So in other words, there was no main effect of survival status on glucocorticoid levels. Another way to say this is that if we held drought risk constant at 0, representing typical rainfall for that time of year, there was no difference in predicted glucocorticoid levels for survivors and fatality, regardless of which season they were compared. So to recap these main results, yes, to answer a question, individuals with stronger positive fecal glucocorticoid responses, so those that responded more strongly positively to increasing drought risk, those females were more likely to survive the severe El Nino drought. And this finding remained even when we controlled for the potential confounds of age and rank. Now I'm gonna pass along audio to Sophia.
Great, thanks. So why do some capuchins appear to have a better response than others? So as Irene mentioned, first, we found no evidence of HPA axis dysregulation. This is just another way to visualize that. During the wet season, which is what we were essentially calling a baseline, they showed the same glucocorticoid levels, and then they also showed that they both showed the expected increase in response to the dry season. Next. So there are a few different reasons beyond HPA dysregulation. First is context-dependent evolutionary selection. So the same trait can have diametrically opposite fitness consequences in different settings. Next. And so we can have selection for relative fitness that maximizes fitness across all environments, across the most common environment, or across the most harsh environment. And this was a particularly severe El Nino. Remember, it was one of the 3 most severe ever recorded. So it's possible that having such a strong reaction to drought wouldn't have been all that beneficial to less severe instances. And finally, there's lots of interest in how developmental experiences in utero or infancy can alter lifelong stress responsivity in adaptive or non-adaptive ways. And one of our co-authors, Ashley Mensing, is exploring this for her dissertation. Next. So quickly, just going over the importance of this study, we're really excited because this shows some of the first evidence from wild primates that a more robust stress response is associated with increased fitness. Next. This provides really encouraging results and advocates for using a within individual or reaction norm approach to hormone research in primates, in the wild, in the lab, in natural settings, and even in humans. And finally, El Nino events are increasing in frequency and severity, and these have global effects on rainfall and temperature. So it's crucial for us to deepen our understanding of how these natural disasters impact the health of human populations and the conservation of wild animals. And with that, I'll pass it to Susan.
So I've already told you a little about these co-authors. Maybe I don't need to say too much here then. I do want to thank specifically Colleen, who really kind of got our behavioral endocrinology work started here. I think she's in the audience today, and Julianne Dam, who worked with Colleen in the early days of that and is still doing behavioral endocrinology work. So she and Colleen and Irene both all contributed a lot of their extra samples to this project. And of course, if you want to pull up the next slide, so many people to thank in a long-term study. We had 124 field assistants. I think it was, dozens of thousands, probably close to 80,000 hours of observation just for this segment we talked about today. Many, many, many grants I had to write, and other people wrote too, Irene in particular, and Colleen. And thanks, of course, to the Park Service and to the private landowners who let us work on their land as we were collecting these observations and samples. And yeah, I'd like to open it to questions now.
Thank you so much, Susan, and everyone. This is just incredible. If we could stop the screen sharing for a moment there. There's going to be lots of questions. I think we should call on people in the order that they put things in the chat. Kyla, I think you'll be first if you would put up your hand so we can find you easily. But first, just imagine 80,000 hours of data collection compressed into an elegant 25-minute presentation. Wow, I mean, this is just amazing. Kyla, why don't you go ahead and unmute yourself and show your camera if you can.
1 second, please. Okay, hi, my name is Kayla. I'm a graduate student at East Carolina University, and I want to say thank you for this awesome talk. Immediately, what was coming to mind when you were discussing the shocking lack of maternal investment with these infants, I was thinking about other social dynamics of capuchins, because my understanding is they're fairly social primates. So did you see any other trends in terms of like less grooming, copulation, cohabitating? Were groups splitting? So what other social differences were observed?
Yeah, so we didn't see any group fissions. It's sadly this this corresponded also to a funding drought and a personnel drought. So at this point in time, I was trying to monitor 15 groups with 3 people, and we have 2 people per team. So really that meant we could see 1 group per day. It also corresponded to the time when our car was totaled and we had to walk 2 hours to the site and 2 hours back at the end of each day. So it was much worse quality monitoring than usual for us. And sadly, we didn't get enough focal samples, enough focal density to do a quantitative analysis of the behavior. So I'll have to rely on, what I remember, what struck me when I was there during this period. It did seem like the inter-individual distance was a bit wider than usual. They were having to eat really crappy foods then, and so, stuff that they normally wouldn't touch. They were grooming. They maybe not as much, but again, I can't really pinpoint the exact differences. It wasn't a dramatic decrease in grooming. It was really the maternal behavior that set off the alarm bells, and that was completely outside normal for capuchins. But yeah, they were still mating. They were still doing most of their normal capuchin stuff.
Any other comments from anybody else in the group before we go to other questions? Any others? So I'm looking for hands being up. So there are a lot of good questions in the chat, and I hope people will raise their hand. Looks like Wendy has, let's see. Bernie, you're next. If you can unmute yourself and go ahead.
Right, yeah, thank you. That's really amazing work. I'm just wondering about whether you have the data that would allow tests for survival reproduction trade-offs. So, for example, did the females that died, did they have higher age-specific reproduction than the females that survived, that could provide insights into the costs of the stress reactivity being high?
I mean, we do have, of course, data on reproductive output, but we haven't done that analysis, so I don't think I can respond right now to that. They were having higher infant mortality rates at the time of the drought, but I don't know whether in general they did.
I think Santa Rosa has some data on not as severe El Niños necessarily, but that generally I do think there is an impact on infant survival. So I think there's some indications that less severe versions have an effect, but yeah, we haven't analyzed specifically the infant or sort of reproductive output costs with the drought.
I guess the question would be, why aren't they also more stress reactive? What at least the potential costs of stress reactivity, maybe something besides reproduction? It's really variable, that's all.
Mm-hmm. Yeah, we've been puzzling about that too. We don't have a good answer yet.
Good. Next, let's go to Barbara Natterson Horowitz. You unmute yourself and turn on your camera there, Barbara.
Okay, sorry about that. I'm not on camera right now, but thank you. What an interesting study, and very really makes the case for moving beyond the way the sort of singular demonized view in which stress is so often portrayed. So it was so interesting. My question is whether we know what the causes of death were of the juveniles and the older individuals, and specifically whether you feel that the adaptive benefits of the stress response were directly related to the calorie scarcity. So the gluconeogenesis and the all the ways in which glucocorticoids liberate energy, or related to some of the secondary effects of calorie scarcity, including maybe vulnerability to parasitic infection or alterations of social structure.
Yeah, I mean, we didn't find bodies, and that's common in a tropical forest. Bodies decompose within a couple days, or they get picked off and eaten by carnivorous wasps and vultures and so forth. But the ones that did disappear, who we assume died, were very, very thin when they died. So we do think that this was an outcome of nutritional stress. We don't have any other physiological markers, though, so we can't provide a super satisfying answer to that.
Yeah, and to address the second part of your question, I think it would make more sense for the stress response to be relating to anti-starvation mechanisms, as Susan mentioned. And so we know that maintaining high levels of cortisol and other glucocorticoids is going to actually reduce immune function. So if it were the opposite, we would actually expect those individuals to potentially have a greater risk of dying from vulnerability to parasites or other diseases.
Let's go next to Michelle Blythe. Michelle, could you unmute yourself and turn on your camera?
Hey, it's Michelle. Thank you for that really interesting talk. This is a little bit outside of my specialty, but in humans, at least, it's not entirely uncommon to have things like maternal diabetes or just maternal elevated glucose. And I was wondering if there might be any sort of link to that with these glucosteroids being elevated in stress and all of that.
Yeah,
we don't really have go ahead.
Sorry. Yeah, I mean, it's not happening in wild primates, but definitely in humans, there's links between chronic stress and numerous diseases, but it's associated with high blood pressure, which then and the development of diabetes eventually, and that all makes maternal diabetes during pregnancy or preeclampsia more likely as well.
Really appreciate the clear questions and short to the point responses. This is just wonderful. Melanie, if you would unmute yourself and turn on your camera, that'd be great.
Hi, thank you for a great talk. I was wondering if you have looked at evidence for dysregulated cortisol in other contexts. So here you're seeing like what you expected. This kind of follows up on Barbara and Bernard's question, but you're seeing that expected rise and then greater survivability for those who had the highest rises, where it seems very advantageous. But what about in other stressful, extreme stress contexts that might, in particular, like psychosocial contexts? Are you seeing evidence of dysregulation, or even among these individuals where higher GC in this context is adaptive, but in another one it isn't, or very low GC, something like that?
No, I don't think we've done that yet. I mean, we have, Colleen long ago did look at the FGC levels and how they change according to social stressors, but we didn't look at it in exactly the way you're asking about. So yeah, I don't think we can answer that yet, though probably when Ashley's done with her thesis, we will be able to answer that question.
Okay, thanks.
Oh, Nick Thompson
sorry.
Gomez, please go ahead.
I'm going to step in for a second.
Go ahead.
Just to answer that question a little bit better, if we had to make a prediction, we shouldn't see dysregulation. These are adapted animals to their environments. Unless we have severe anthropogenic, well, climate change, obviously, but if we do something where we're losing forests and they're losing food, possibly. But the theory predicts that animals should be adapted and the stress response shouldn't ever get dysregulated. It's a flexible response. And in the wild, we actually have there's a lot of data that show while chronic stress might exist in the sense that it's long-lived, it's not chronic in the same way it is in humans. So in wild animals, we expect that the cost of not doing a response is going to be way worse than the cost of doing the response. So it's always going to be better to have this glucocorticoid response in response to stressors than not.
Thanks, Jacinta. Actually, I'm going to tuck in my big core question here and ask Susan and Jacinta and the other authors. What do you think is the essence of the trade-off here? I mean, what is it that makes the cortisol response so expensive? And what can you just summarize what the trade-off is? Maybe you want to go first, Susan, and then Jacinta or others. Go ahead.
Actually, that seems like more of a Jacinta type question.
Okay.
Do you think?
Sure. There's no free lunch. Like, when you are doing this expensive process, you have to pull the energy from somewhere. We have catabolic processes and we have anabolic processes. And so to pull, to basically do this response and to have this extra energy that will get you through the drought, there is going to be a toll taken from somewhere. Generally, we don't see this until further down in an animal's lifetime or a human's lifetime, like those baboons at Amboseli, the ones that were kind of bathing in glucocorticoids across their lifespan, they died 5 years earlier. That's real. Like, that is definitely the wear and tear that Sophia mentioned that causes us to, take it's a toll, but it's a toll down the road. And if you don't get down the road, you never see that toll. So the question is now, in terms of looking at, what's better, you need to survive that lion. You need to survive that drought. So you need to kick in that glucocorticoid response every single time you have a chance to survive that immediate stressor so that you can live to maybe later have a wear and tear. I think the problem with the way we've approached this, me included, the problem with which primatologists and field biologists have approached this problem is we look at population-based glucocorticoids. And we're comparing apples and oranges. We're comparing a group that has few lions to a group that has a lot of lions. And so if the group with a lot of lions dies, that's because they have a lot of lions. It's not because of the glucocorticoids. And so, this is an idea that Robert Sapolsky popularized in his book, Why Zebras Don't Get Ulcers. But basically, this idea is that we need to measure those stressors. Yes, there we go. Thank you, Megan.
Showing the book here.
Yeah, no, it's a great read.
So thanks so much, Jacinta. Because this is so central, do any of the other co-authors of the paper have brief comments you want to add to this?
Yeah, well, I would just relate it back to humans as well, given this is Club Ev Med. So in humans, we are unique in that our stress response is activated in response to things that are not actually life-threatening. So going to work, giving a presentation, we are activating that stress response, and doing so chronically is what might eventually lead to dysregulation. So just remembering to keep that in mind. Definitely wear and tear, and seeing that those negative effects of chronic stress is a lot more common in humans and makes a lot of sense, but we don't really get the chance to examine how the stress response itself in response to a particular life-threatening stressor is actually beneficial.
Well, thanks so much, everybody. Let's go on to Negan's question here.
Thank you. Hello.
Sorry,
Thank you for
Randy.
the-
I was just about to ask a question when you
Oh,
had
yes.
jumped in. Could I
sorry there. Go
finish
ahead.
that one? Thank
Okay.
you so much. Hi, everyone. Thank you guys for a great presentation. I was curious if you guys have had resolution of like temporal resolution in your sampling to be able to examine something about the resolution of the stress response and see if there's any relationship with the ability for an individual to return to its kind of starting state. I won't even call it baseline. If that corresponds at all with the strength of the response that you're seeing as advantageous in your study, et cetera.
So you mean more like a time series analysis where you see they've been exposed to drought risk, and now the drought risk has been has dampened and it's back to normal conditions, whether then we would be able to see that they go back to their baseline levels.
I think that the broader importance of just the broader significance of the question is just we kind of know that it's important to be able to bring yourself back down to normal after having a response, to not slide into what would be a chronic or maladaptive elevation of glucocorticoids. And so, there's a kind of there are 3 points to the cycle. I'm just wondering if you guys have looked into the resolution of the stress response.
Yeah. So just unfortunately with the data we have, we could only look at the first half, right? The increase from normal levels to stress induced. And essentially it was fecal samples, which, are arguably already showing some sort of average over time, and then just not a ton of samples collected sequentially for a female, like all within one season or something like that. We had to combine data across 5-6 different years for this.
So we might be able
Okay.
to do more with that in the future, in that they're a subset of these monkeys. We do have high density thanks to Colleen and Irene doing their work. But then, of course, it depends whether you mean after a small drought or the big drought, because, of course, after the big drought, a lot of them are dead, and so they do not continue to poop. We do have a lot of not yet analyzed post El Nino samples from the survivors, where we could someday go analyze those and see if we did sample intensively during that period and have not yet looked at those samples. We haven't measured them yet.
Thank you. Yeah, it's really exciting to just get deeper into what is a good stress response. So that's all I'm asking. Thanks.
Yeah.
I think Colleen has a comment there, which would be great
So
to.
we have
could you call
5.
Colleen? Because she is a co-author and has
Okay,
thought about
Colleen,
a lot of
please.
these more than us.
Please, we can't find people unless they raise their hands. So Colleen, please go ahead.
Hi, so can you hear me?
Yeah.
Yep.
Okay, great. So to address that question more directly, I actually tried to look at the resolution of the stress response with these monkeys using fecal samples. And so I did that by following the same individual for 2 to 3 days in a row and documented what they experienced, both social stressors and arguably physical stressors, like when there was an intergroup encounter and they had to flee or fight. And I tried to look for actually basically the shape of the curve of the response within individuals, and was able to see that pattern. But the lives of these monkeys are just incredibly dramatic. They love drama. There's always a lot happening, and to be able to relate all the different potential sources of stress, be them environmental, physical strain, social drama, being a mother, being pregnant, down to what was actually happening on the level that we can see with the fecal samples. It was just incredibly complex. And I was only able to really see individual events that they were responding to when it came to something like an intergroup encounter that was a combination of different forms of stressors. So I would say that it's not impossible to get down to that resolution, but definitely limited with fecal samples.
I think it's also part of that challenge is how hard it is to get the consecutive daily samples. So when I was collecting for trying to extract progesterone levels to see when females were cycling, you could very quickly within a day see a rise and a peak, and then you'd want to see when that went back down again. But just missing 1 or 2 days of sampling, because these are arboreal primates, right? You can't always catch when they defecate. It really increases the challenge of being able to do such a fine temporal resolution. It's just really hard to get 5 consecutive days. And when just missing the 1 or 2 days really can set back that entire attempt to get a return back to baseline.
I just want to say that Colleen's 3-day focal follows should have been an Olympic event. I mean, it was amazing how hard it was to do, and it was just awe-inspiring that she did it.
Wonderful comments. Thank you, everybody. I see we have 5 hands up in 5 minutes. We may not be able to get to everyone. Megan, you had a nice question there. Please go ahead.
Sure, yeah, thank you. Thank you for the great talk. I might misunderstood that part, but I thought that in the first part of the presentation, we talked about, how stress is bad for you and it can cause like diseases in humans. It also kills like higher, I think, cortisol levels will kill capuchins up to 5 years earlier. But in the second part of the, the presentation, we're actually reviewing the results. It seemed that the stress response is actually helping them to survive. So I wasn't sure if these two were conflicting or were actually different parts of the equation. Could you a little bit clarify?
Yeah, sorry, I'll take a stab at that. So the first part, so that actually wasn't a study from capuchins. That's a different study by a different group of researchers from the Amboseli baboon project. And yes, theirs is a very clear example of the wear and tear of having high cortisol throughout your life is going to eventually catch up to you and lead to probably shorter lifespan. But they were not looking specifically at how one individual's response to a specific stressor, whether it allowed survival of that stressor. It was just looking big picture. These individuals have high glucocorticoids, these have low, and these ones die sooner. Does that make sense?
Yeah, maybe a longer term also, I guess, aspect of it, rather than, okay, now you're in a tough condition. If you have higher cortisol levels, maybe it may help you actually survive because it affects your actions too. What you do to survive, right?
Yeah.
Okay.
So we're really focusing here in this study on the immediate survival of the immediate cause of the stress response.
Okay, cool. Thank you.
Okay, Ned, could you unmute yourself and show your camera there?
Sure. A heroic study and beautifully presented. Thank you. I know we've focused a lot on the scarcity of food. There hasn't been much attention paid to the scarcity of water during an extreme drought. And so it has me thinking as a part-time endocrinologist about the aldosterone. And so Jacinta could probably reply to this in terms of, I wonder if the elevated cort in the survivors is perhaps a reflection of a really robust aldosterone response, since corticosterone is a couple of steps above it, and maybe your fecal metabolites for glucocorticoids is also picking up corticosterone metabolites.
I actually want to jump in here and say they did not have a water shortage because they have all these permanent rivers running through this. And so that would not have been a factor in this study.
Oh, great. Thank you.
But I like how you're thinking.
Yeah.
We can't separate cortisol from corticosterone in our fecal samples. We have to do a generic glucocorticoid assay. So we're unable to determine that. But it is a great thought, and one that certainly would come into play if we were dealing with water versus food. Thank you.
Thank you.
Yeah, one of the other long-term studies like Santa Rosa, they do have more limited water, and so that would be a better place to investigate something like that.
So Charlie, I think we're at the top of the hour. This is a wonderful discussion. I think we should probably stop, but if you'd like me to carry on for 1 or 2 minutes with these short things, we can do that. What do you think?
Yeah, I'm starting to see the numbers drop a little bit as we get to 1 o'clock here on the East Coast. I guess we could do one more question if we want to do that, but then we should probably wrap up.
Mandy's got a very straightforward, important one. Why don't you go ahead, Mandy?
Thank you. Great talk. Yeah, I was just trying to make this quick. I was reflecting on my own experiences studying Kinda baboons, and we had a very similar situation with irregular rainfall and lower availability of food and everything like that. And I did visibly see them get emaciated, like Susan was describing. But I was curious because around the time that was going on with my Kinda baboons, we had a higher ranking young mother. It was her first infant. She lost that infant and she carried it around, the deceased infant around for like 10 days. So, and from my other experiences with Hamadryas baboons and everything, I realized that like sometimes it's the younger mothers that are just not as great at mothering because they're still learning. So I was just wondering if you observed any of that in the capuchins, like with the infant mortality. Did you see infant, like younger adult mothers lose infants more than the older adult mothers, or is it strictly just the glucocorticoid levels influencing that?
During non-drought conditions, you do see more of young mothers losing infants, just lack of experience, I think. But during this drought, everybody, just about everybody was losing their infants, including the very old.
Wow. Okay. Yeah, that's great. That's so interesting.
Yeah, it
Thank
was a
you.
terrible, terrible mortality event.
Yeah, yeah, so
Yes.
sad.
Okay, thanks everyone for really a stimulating discussion. Charlie has posted on the chat that the next one is going to be April 23rd, A Challenge to Pedo's Paradox by George Butler and Chris Vendetti. We hope to see you all there. Go ahead and sign up at clubevmed.org if you if you don't get emails from us otherwise. Wonderful discussion today. Charlie, do you want to say any wrap-up things and say goodbye to people?
Just thank you all for joining. Thank you, Susan. Thank you, Irene, Sophia, Jacinta, other members of the team for sharing this really exciting paper. And just really appreciate the great discussion we had. Thank you so much, everybody.
Thanks, everybody. Bye-bye.
The invitation.
And thank you, Randy.
Yep, thanks.
Bye.
Okay. Bye, everybody.
Bye.
Thank you. Bye.